Welcome everybody to this international symposium on stroke recovery, which is organized by a joint symposium of the OHBM and the DGKN, the German Society for Clinical Neurophysiology. The idea of this symposium is to connect the high expertise in methods in brain imaging in electrophysiology, which is available in the OHBM community, with the clinical expertise provided by the DGKN. And that's why we have organized the symposium consisting of representatives of these two organizations, which is Heidi Johansen-Berg and Steve Small, and two from the DGKN, which is Friedhelm Hummel and myself, Grefkes. Well, I'm finished now.
Yes, I got that.
Or should I have some like a closing remark so that I now start with the first talk, or what would you suggest?
Actually, it's a good introduction.
Good. I will now start with the first presentation, which is my talk, entitled Cortical Reorganization After Stroke, Novel Insights from Neuroimaging and Non-Invasive Brain Stimulation. Stroke is a devastating disease because it has enormous impact on quality of life and disability. It is worldwide the most important cause for an acquired disability, and more than 50% of people surviving a stroke keep a permanent deficit. And for these patients, recovery of function is the most important mechanism of regaining functional independence and quality of life. And an important driver of recovery of function is a neural reorganization. And we can image neural reorganization by means of functional magnetic resonance imaging. For example, in the motor system, when healthy subjects move their right hand, you see that motor cortex activity in the left hemisphere is mostly activated in a very lateralized manner. When the same task is performed by stroke patients suffering from a right hand motor deficit because of a left hemispheric stroke, you see that brain activation patterns are greatly abnormal, not only in the lesioned hemisphere, but also in the unaffected hemisphere. And now the question arises, what is the meaning of these changes in activity? Is it supportive? Is it learning associated? Is it maybe even maladaptive, disturbing? And to address these questions, neuroscientists in the past 20 years have used imaging and also brain stimulation approaches to disentangle different mechanisms engaged in recovery. And recent development, for example, in connectivity, now allows us also to look into temporal aspects of fMRI connectivity. This is referred to dynamic connectivity, which uses a sliding window technique and then can show how networks spontaneously form and then disappear. And this fundamental property of the human brain is greatly disturbed in acute stroke, which is a network disease and not only affects local neural processing, but also in the entire network. For example, if we do a classical analysis of resting state connectivity in the motor system, we see a decrease connection within the motor system, which is well known and has been shown by many, many other studies. However, when we look into the temporal domain using dynamic functional connectivity, then we can show that there is a strong relationship between the connectivity patterns which are preferred by the patients and their degree of motor deficit, such that patients with severe motor deficits prefer a connectivity state which is characterized by a strong spatial segregation with activity or strong connectivity in the cortical sensory motor network, while patients with mild deficit show a more integrative connectivity pattern. That means already in the first days after stroke, and these patients are within the first 7 days after stroke, connectivity patterns differentiate depending on lesion severity or symptom severity, which may have an impact on outcome and also on therapeutic decisions. However, also in a longer temporal perspective, meaning days and weeks and months, we know that stroke-associated changes in brain activity are very dynamic. Especially in the first 2 weeks, we see an increase of neural activity in both hemispheres, which then, when patients recover after 4 months, go back to a normal activation pattern. Similar things can be observed in connectivity studies. However, when you then ask the question, which connections are most important or most indicative for good recovery, then it's the increase of connections between premotor cortex and supplementary motor area with the ipsi lesion of primary motor cortex, while those patients who develop a negative influence from the motor cortex of the unaffected hemisphere towards the motor cortex of the lesioned hemisphere are those patients who make less successful recovery. And these changes and effects which we observed in dynamic causes modeling of fMRI nicely fits to the classical model of post-stroke interhemispheric inhibition, which assumes that a lesion in the brain leads to an imbalance of the hemispheres with respect to mutual inhibition. And therefore, one therapeutic concept is to either reduce overshooting activity in the unaffected hemisphere, or either to increase activity in the lesioned hemisphere. And one way of achieving this is using transcranial magnetic stimulation. And we have used this method in the past to focally modulate, especially function of the primary motor cortex in both hemispheres. And what we find in combination with imaging is that you can indeed correct pathological network activity and connectivity when you use inhibitory paradigms on the contra-lesional motor cortex. And these normalization of activity and connectivity are associated with a normalization of hand motor performance. Similarly, when you increase activity in the lesioned hemisphere, especially in the very early phase after stroke, you can also improve motor recovery in patients, which is associated with normalization of connectivity in these patients. However, the motor system does not only consist of one, but also other areas are important to maintain motor function. But much more less is known about their role in recovery of function. So we can use, for example, online TMS to interfere specifically with brain regions to infer their causal role while doing a task, as shown here. You can see that this technique can directly interfere with neural processing and interrupt an ongoing task. And this makes this approach very nice if you want to see the differential roles of different areas within the same patient for a given task. And we did this with respect to ipsilateral motor areas, so motor areas or motor-related areas in the unaffected hemisphere for hand tapping performance conducted with a stroke-affected hand. And when we disturb a motor function in these areas, we see quite differential effects. For example, if you do this in the first week after stroke, we see that disturbance of activity in anterior intraparietal sulcus, AAPS, leads to an improvement of hand motor performance of the stroke-affected hand. While we do not observe an effect for M1 interference or dorsal premotor cortex interference in the first week after stroke. This is different 3 months after stroke. Here we see that there is still a disturbing effect observable from anterior interparietal sulcus. However, we see also an effect when we interfere with dorsal premotor cortex activity. Here, patients now show a worsening of motor performance when we disturb the dorsal premotor function. So this means that we have a time-sensitive and region-sensitive effect with respect to recovery. Here, anterior interparietal sulcus seems to disturb motor function already early after stroke and maintain this disturbing role, while contralesional dorsal premotor cortex seem to develop a supporting effect later in stroke. And these differential effects have, of course, strong implications with respect to planning or conducting or using new intervention targets using TMS or TDCS in order to support recovery of function. Finally, I would like to close my talk with presenting novel readouts of cortical activity per stroke. And here I think a very promising approach is the combined use of EEG with TMS. Because this method allows you not only the effect of TMS on the stimulated brain region, but also the response of interconnected brain regions. And because EEG has a very high temporal resolution, you can get this rich picture of network oscillations activating the entire brain in both hemispheres. And if you apply this in a network disease like stroke, we of course expect significant changes of these patterns. And this is shown here. This is a stroke patient 7 days after the insult, which this patient has a severe deficit. He's hemiplegic on the left side. We now applied TMS EEG to the motor cortex in both hemispheres and see what happens compared to the unaffected hemisphere, which is shown on the left-hand side. So now the TMS pulse is administered now. And you can see that on the left-hand side, you have a complex temporal response, while on the right-hand side, you have a very focal, static, abnormal response. And of note is that in this patient, the primary motor cortex, the lesioned hemisphere, was not structurally affected. So here's the lesion, which is more in the corticospinal tract, implicating that this is a disconnection effect, which we can observe in this acute stroke patient. And we know when we do it in a bigger sample of patients that these effects are not only related to the motor deficit, but also to recovery in these patients, such that patients having a more complex temporal structure in this TMS evoked potential are those patients who make better recovery compared to those patients showing this abnormal static response in the very early phase after stroke. And this is applicable or already in individual patients, as I've shown you here in my examples. So this might serve as another readout of network disturbances and maybe will be helpful in the future also for planning of therapeutic interventions. So I would like to conclude that I hope that I could convince you that connectivity models enable a deeper understanding of stroke deficits and functional recovery. We have more and more evidence that contralesional brain areas have a time-specific and region-specific interference effect with motor recovery. And there are novel electrophysiological readouts like TMS EEG available, which in the future might enable individualized outcome prediction and therapy planning. I thank my collaborators and I thank you for your attention.
Hi, everyone. I'm sorry I can't meet up with you all in person, but thank you for coming. Thanks to Christian for inviting me to present. And I'm used to interacting with you all during my talk and looking at your faces and responding appropriately. And I apologize in advance if I can't do that. I'm going to talk today, I'm the language guy. I'm going to talk today about dynamic network changes and language recovery after stroke. And let's get started. There are three basic principles that I'm going to organize my talk around. The first is that human language evolved from the sensory, motor, emotional, and social functions of the primate brain. That human language performance involves multi-system interactions across complex spatial and temporal representations in the human brain. And the spatial encodings for language do not localize to one or even a few regions, and the temporal encodings do not occur on a single timescale. So evolution of language from sensory, motor, emotional, and social functions of primates. Let's look at the primate motor system, the non-human primate motor system. In 2001, there were already more than a dozen, maybe almost 24 different motor regions of the cortex that had been described in macaque. And this was very, very important because subsequently many of these regions have been described using our imaging technology in humans. Doctors Leslie Ungerleider and Mortimer Mishkin in 1982 mapped out two different visual pathways in the macaque. One they called a what network, which was a pathway that was occipitotemporal, a so-called ventral pathway. And the other was an occipitofrontal pathway that they called a where pathway, and has subsequently been thought of sometimes as a how pathway for visual, high-level visual processing. And in 1999, Josef Rauschecker and his colleagues took the work of Ungerleider and Mishkin, in fact, with the collaboration of Mishkin, and mapped this same concept onto the auditory system. And described two different pathways in the auditory system, a what pathway that again is a more ventral pathway, and a where pathway that again is a more dorsal pathway. So my colleagues, Ina Matthias-Schlasewski and Josef Rauschecker himself, developed a concept of the neurobiology of language based on both these multi-pathway models and also the evolution of the brain from the primate. In the world of language and language understanding, using the primate as a model for human language to some people hasn't made sense because, of course, non-human primates do not use language. But for us, understanding all of the multi-sensory and motor inputs into the processing of language was the most important aspect. And so we took the work of Michael Petrides and his colleagues from the Montreal Neurologic Institute, mapping out various white matter pathways for language. And we applied the Rauschecker and Ungerleider approach and said, okay, some of these are in fact ventral pathways, and some of these are in fact dorsal pathways. So from this multiple approach, we move on to our next concept, which is that human language performance involves multi-system interactions across complex spatial and temporal representations in the human brain. In order to do language processing in the real world, that is to say, in the movie theater, in conversations, on the street, in your home, things that we're not doing so much in person right now, involves multiple different kinds of sensory motor processing. And in order to do real live understanding of language requires many of these different kinds of skills. And this was pointed up to us early on when we said, how can we study language in the human brain, normal language in the human brain? How can we do that in an ecologic way? That is to say, not making decisions about what we're seeing in language, not doing meta language, but doing real language. And so we actually studied people looking at movies, and we found, of course, much of the brain is involved in that behavior. So we actually came to the view that the language of the human brain include Broca's area, for sure, Wernicke's area, for sure, but in fact that the language areas of the human brain are really throughout the entire brain, and that virtually the entire brain is involved in language processing, and including subcortical areas and cerebellum, which are not shown in this caricature. To connect all the different components of the brain that are participating in language comprehension, we need a bunch of communication pathways. And although we were focused on two broad categories of these pathways in the dorsal and ventral portions of the brain, in fact, the number of these connections is very massive throughout the brain. And there's much to learn about connectivity, both functionally and anatomically. So the third component of my short presentation today is that the spatial encodings for language do not localize to one or a few regions, and the temporal encodings do not occur on a single time scale. So the fact that connectivity matters for stroke recovery is not something that I invented. It's not something that's been invented in the last couple of decades. Ramon y Cajal pointed out the complex connectivity structure of the brain, but von Monikoff in 1914 noticed that the complex connectivity structure of the brain was actually relevant for stroke recovery. And finally, more recently, Nina Dronkers and her colleagues have analyzed the first patient of Pierre-Paul Broca and analyzed this patient in a way that Pierre-Paul Broca couldn't have done. Now, Broca never even cut this brain, so he didn't know what was inside this brain. He saw a lesion in the inferior part of the frontal lobe, left frontal lobe, but he did not cut this brain. What Dr. Dronkers and her colleagues did is to do magnetic resonance imaging of this brain and found that there was much more damage to the brain than would be seen just on the surface. And in fact, connectivity throughout this brain was massively impaired. So building on all this work in normal language and the normal structure and function of language in the brain, my colleagues Giovanni Buccino and Anna Saladkin and I developed an approach to the study of stroke recovery built on connectivity and built on evolutionary principles. And in fact, our work in this area has focused on one such aspect of the overall brain architecture, and that is the action understanding system of the brain, the so-called mirror neuron system. And to take a look at my little animation there, and also the role of the dorsal stream. So this work has mostly been done by Susan Duncan with some help from Priya Santhanam. Susan was a PhD student and Priya was a postdoctoral student in my laboratory over the last decade. And what we ran, the study we ran was a study of action observation therapy, that is targeting the action understanding system of the brain. And we had 19 subjects, and you can see on the left of your screen the lesion overlap map in the 19 different subjects. We did therapy for 6 weeks, but we did a lot of imaging. We did imaging 6 weeks before therapy. We did imaging 6 weeks after therapy. And in fact, in each imaging 6-week period, we did 3 separate scans. So basically we have scans on each one of these people at multiple time points during their before therapy, during therapy, and after therapy. I should point out, go back just to point out that yes, there was therapeutic benefit to the action observation therapy. The reason I actually went by that so quickly and wasn't even going to pay any attention to it is because the therapeutic advantage was not significantly better than any other therapy that had been done over the past century. That the therapeutic advantage had a very small effect size. But what we did notice some things behaviorally, and we did notice some things in imaging. Behaviorally, what we noticed is that performance variability of these people related to their improvement. And the more variable they were before treatment, the better their improvement. Furthermore, their variability went down as a result of therapy. So behaviorally, we see that there's something about certain behaviors that are susceptible to our therapy. The first thing we noticed about the biology of this is that the group of 19 was not homogeneous. Now, of course, no group of stroke patients is homogeneous. We've learned that over many years, and particularly in the 20th century, this became very clear to many workers in language particularly. In our group, it turns out the patients with moderate term injury, that is the ones who had only had their strokes within a few years, actually got some reorganization in the left hemisphere in the connectivity pattern, and you can read about that in some of these papers. With longer term injury, that is, they've had a longer time since their stroke, they had more reorganization in the right, and there's a little bit less to show for that actually in that right-sided reorganization. Finally, Susan Duncan and I and our collaborators, we looked at the resting state networks in all these stroke patients, these people with aphasia. And these are 8 of the resting state networks that were present in this group of 19 people. And you can see that these resting state networks look pretty similar to the ones that occur in normal people. And in fact, if you look at the paper that's cited there first, Van den Heuvel and Hulshoff, they show the resting state networks in healthy controls. They look something like this. Of course, if you look at image F, there's really no component in the right hemisphere in this particular resting state network because of the presence of the stroke in that network. Those of you who know graph theory a little bit as it applies to imaging know that one of the main one of the main measures of graphs and graph segregation integration is a measure of modularity. And in our patients, increased modularity of the resting state networks was actually associated with better outcome. CIU there is content information unit, and that's a measure of semantic processing, a meaning processing of these words. And so we had a better change in meaning comprehension with people who had more modularity. And the last thing I'm going to go through is the time-space analysis, because what I mentioned and one of our guiding principles is that we're not just talking about spatial reorganization, but we're talking about reorganization of processes over time. And one time-space analysis is dynamic functional connectivity. And in doing dynamic functional connectivity on the resting state networks in these patients, we found actually 10 different states. The first 9 are in the top right corner of your screen. Those we found to be interesting and show certain properties that you can read about. The one I'm going to mention that I'm going to talk about a little bit right now is state number 10, because state number 10 is a state of this resting state process that it shows maximum separability across the classic resting states. So state number 10 is different. It differs from all the other resting states, and it does so maximally. So I guess one take-home message here is that the resting state itself over time is a bunch of resting states. And if you look at the state, certain states can show different properties than others. And so the last point is that those patients, those participants who stayed in resting state 10 the longest actually had the best improvement in their semantic processing, in their change in content information units. So that's all I have to say. But in summary, I want to say that evolution is critical, our guiding principles again. Evolution is critical to understanding the neurobiology of language. Language representations are bilateral and distributed. Language functions appear to organize around numerous dorsal and ventral pathways and in many time-dependent states. And all of these properties impact language recovery after stroke. And without apology, I tell you, you should buy this wonderful book on neurobiology of language, and please submit papers to our new open access, open data MIT Press journal called Neurobiology of Language. Thank you very much, everybody. I wish I could see you all.
Hello, my name's Heidi Johansen-Berg, and I'm going to be talking about neuromodulation to improve motor recovery after stroke. So neuromodulation-based adjunctive approaches to rehabilitation after stroke can be used in a couple of different ways. They can be used to try to drive brain activity towards a more optimal pattern during rehab, or they can be used to try to boost or trigger processes of brain plasticity that would be thought to occur during rehab. And one popular neuromodulation technique is the use of non-invasive brain stimulation, such as transcranial direct current stimulation, or TDCS, which is illustrated here. So this is appealing in the context of stroke, because we know that after stroke, brain patterns of activity tend to be non-optimal. So for example, we know that there tends to be underactivity in the stroke hemisphere and potentially overactivity in the non-stroke hemisphere. And TDCS is an attractive way of dealing with this because it provides us with a focal method for the either increasing or decreasing activity in a targeted brain region. So for example, we can apply anodal or excitatory TDCS to the stroke affected hemisphere as a way to try and boost activity in this region. And we've done a number of different experiments with this approach, and these studies have been done with Charlotte Stagg, my colleague in Oxford, who's modeling the TDCS kit here in this picture. So one of the first studies to apply this approach in a proof of principle way was from Friedhelm, Hummel and colleagues some years ago, where they showed in a group of individual patients that you see illustrated here, that when anodal TDCS to the stroke affected hemisphere was being applied, patients' movements got faster. We've combined this approach with fMRI to look to see which brain areas are changing their activity in association with those changes in behavior. What we found in these studies led by Charlie was that if we compare a group receiving real TDCS to a group receiving sham TDCS, we find that the TDCS results in increased activity that you can see here in the stimulated motor and premotor areas of the stroke-affected hemisphere, and that also, as you see on the right there, the degree of activity increase correlates with behavioral gains. So this supports this idea that we can use TDCS as a neuromodulatory technique to drive activity towards a more optimal pattern, in this case, to increase activity of the stroke-affected motor areas. We can also have a rationale for the use of TDCS as a method for boosting plasticity. So there's a body of evidence that suggests that TDCS provides an environment which is particularly receptive to plasticity and learning. So for example, studies by Charlotte Stagg have demonstrated that anodal TDCS applied to the motor cortex in healthy people speeds learning, and that it alters brain activity and brain chemistry in such a way that excitatory plasticity might be more likely to occur. So for example, it reduces local GABA. Other groups, so Rice and colleagues, for example, have shown that repeated sessions of TDCS in healthy individuals lead to cumulative and long-lasting improvements in motor learning. So together, this provides a rationale for using TDCS as a neuromodulation approach post-stroke to test whether repeated sessions might drive learning and brain plasticity and therefore lead to better rehab outcomes. And we've tested this possibility in a randomized controlled trial in chronic stroke patients, the results of which are summarized here, where we found that the addition of anodal TDCS to an upper limb rehabilitative training program gave rise to bigger and longer lasting clinical benefits. So you can see, for example, on the right there, the change in ARAT, one of the standardized clinical scores, in this case, the Action Research Arm Test. You can see that the anodal group in green have bigger improvement and that those persist right out to the 3-month follow-up time point, and that's in comparison to a placebo control, a sham control. In this study, we also acquired brain scans at different time points through the study, and some of those results are summarized on the right, where you can see the fMRI changes. So here we're looking for changes in movement-related fMRI activity pre to post intervention, looking for changes that differ between the anodal and the sham control group. So we find that there's increased recruitment of the stimulated motor cortex in the anodal group compared to the sham group. So this suggests that TDCS as a neuromodulatory technique primes the brain for plasticity, and that has cumulative and long-lasting clinical benefits. For the second half of the talk, I want to move on to a different neuromodulation technique, in this case, MRI neurofeedback. So here we're tapping into the brain's intrinsic capacity for activity modulation. Typical neurofeedback setup is illustrated in this schematic here, where you have a subject lying in the scanner, in this case, performing a task, for example, hand movement. We're reading out their brain activity signals in real time, automatically processing those on the computer, and then feeding back a visual representation of their brain activity that the subject can see. The subject is instructed to modulate that visual representation in some way which corresponds to them modulating their brain activity pattern. So for example, we've used this approach in the motor system, where the objective has been to try to increase the laterality of motor cortical responses during movement. So in this example, we have a region of interest over the contralateral motor cortex in red and the ipsilateral motor cortex in blue, and the amount of movement-related activity in those two ROIs is fed back to the subject through this visual display, where the height of the bars represents the amount of activity in those two regions of interest. The participant is instructed to increase the height of the red bar and decrease the height of the blue bar, thereby increasing the laterality of movement-related activity. And what we found is that both in healthy older adults on the left and in chronic stroke patients on the right, they are indeed able to use that feedback to increase the laterality of their movement-related activity. You can see that in both experiments, the real neurofeedback group have a more lateralized activity pattern than the sham neurofeedback group. So this is encouraging, but obviously for this observation to have clinical relevance, we need to test to see whether those shifts in activity persist once the neurofeedback is taken away, and to test whether they have any consequences for behavior or clinical scores. In both of these experiments, where subjects had a single session of neurofeedback, we found that even though they could use the neurofeedback effectively while it was present, as soon as we took the neurofeedback away, the activity patterns reverted back to baseline. So in a current study, we've been testing whether repeated sessions of neurofeedback can give rise to longer lasting shifts in activity and whether it gives rise to any clinical benefits. So this is a big study that's involved many different people in my lab, some of whom are listed in the bottom right there. And in this study, subjects come in for multiple different scans. So for example, they have a day 1 scan, then they have 3 sessions of neurofeedback on different days, and then a follow-up scan on day 5. They also have a range of different behavioral and clinical measures. We acquire EEG data that I'm not going to talk about today, and they have a 1-month clinical follow-up. So first of all, looking at some preliminary data from the neurofeedback sessions themselves, we can find that, again, stroke patients are indeed able to use the neurofeedback to shift their activity towards a more lateralized response. In this case, we have our 3 different days of neurofeedback, and on each day, there are 3 different blocks of feedback. And you can see that within each day, the real group are consistently increasing their laterality over time, and that comes out statistically. Whereas in the sham group, there's no consistent pattern over time. You can also see that, though, that there's no evidence really for anything changing over those 3 days. So each day looks pretty much the same, and consistent with that, there's no statistical evidence for an effective day here. So even though the subjects are successfully using the feedback within each day to shift their laterality pattern, it doesn't seem to be a cumulative effect over those 3 days. Similarly, we haven't found any evidence as yet for transfer. So if we compare the day 1 to the day 5 fMRI data, there's no evidence that effective shift in laterality that can be evoked during feedback does not seem to be giving rise to transfer effects, so it doesn't persist after the feedback is removed. Similarly, when we look at the behavioral and clinical scores, whether during training or at the 1-month follow-up, there's no evidence that the neurofeedback is giving rise to clinical benefits as yet. However, interestingly, we are finding evidence for effects on white matter microstructure. So on those day 1 and day 5 scans, before and after neurofeedback, we're also acquiring DTI data, which we're using to calculate maps of fractional anisotropy, or FA. And then we're calculating the average FA within the 2 corticospinal tracts, and then calculating a laterality index using those data. And as we would predict from many prior studies, these patients tend to have an asymmetrical FA, so they've got reduced FA in the stroke-affected corticospinal tract at baseline, and that's particularly so in the patients who've made a poor recovery. But we can then look at how that FA asymmetry changes over time and varies between groups, and we find an interaction between time and group, which is reflecting the fact that we're seeing a decrease in asymmetry in the group receiving real neurofeedback compared to the group receiving sham neurofeedback, and that's driven by an increase in FA of the corticospinal tract of the stroke-affected hemisphere. So in other words, it appears that the real neurofeedback group FA is becoming more symmetrical, more normal, if you like. This is particularly intriguing because it echoes results we found in a recent study in healthy adults led by Cassandra Sampao-Baptista, who has contrasted 2 different neurofeedback conditions. The dissociation condition is very similar to the condition I've just described in patients, whereas the association condition, participants are driving up both bars. What we find is that when we look at the neurofeedback conditions themselves, that again, participants can successfully drive their activity in the instructed direction. We can then look to see what effect that has on white matter by looking at the DTI data acquired before and after each neurofeedback condition. And we find a significant interaction in the cluster here, the red cluster in the corpus callosum here, and the data that underlie that interaction illustrated at the bottom there. And what you see is that in the real neurofeedback group, we're finding an increase FA in this cluster during the association condition and a decrease in FA during the dissociation condition compared to no change in the sham control group. So in both of these studies then, it seems that effective activity modulation triggered by neurofeedback does give rise to changes in white matter microstructure. So to summarize then, in a rehabilitation setting, neuromodulation can be used to drive activity towards a more optimal pattern, or it can be used to boost or trigger processes of brain plasticity. Brain stimulation boosts plasticity and enhances rehabilitation outcomes. Neurofeedback, in our hands, does appear to shift movement-related activity towards a more lateralized pattern, but as yet we haven't found evidence that this persists after feedback is removed, or that it results in clinical gains, despite this intriguing finding of microstructural changes triggered by that activity modulation. So finally, just like to thank all of my group members, collaborators for the previous TDCS study, and then current group members who are currently participating in the neurofeedback results that I presented to you. Thank you very much to my funders, and thank you very much for your attention.